A vertebrate stopped its heart, stopped breathing, let ice crystals fill its body cavity, then walked away. That is not a metaphor. In 1982, Kenneth Story at Carleton University published the first confirmed documentation of a vertebrate animal surviving complete cardiac arrest. Not slowed function, not hibernation, not reduced pulse. Zero. A frozen frog, rigid as a stone, no heartbeat, no respiration, no measurable brain activity. Then, when temperatures rose, it moved. The frog was common, found across North America, a species most people step over without a second thought. That paper forced the first crack in what physiology considered a hard wall. A vertebrate cannot survive without a heartbeat, cannot survive ice forming inside its cells, cannot survive without oxygen to the brain. Every one of those rules existed for a reason. They had been tested, confirmed, and taught for decades. Then, Story's data sat on the page and refused to comply. He was not the last. 5 animals are filed in this record. Each one survived a condition that, by published physiological limits, should have been immediately fatal. Not dangerous, not damaging, fatal. Each survival event is tied to a named study, a specific temperature, a specific date, a specific researcher who had to reread their own numbers. An Olm salamander in a cave in Slovenia, a sea cucumber on a Caribbean reef floor, a painted turtle sealed under 4 months of ice, a wood frog in a frost-covered forest in Ohio, and one more. John Costanzo at Miami University published findings in the Journal of Experimental Biology in 2014, documenting survival at -16 degrees Celsius for multiple weeks. His quote, "Ice formation is normally lethal to cells." Normally. That word is doing extraordinary work. Before this ends, the mechanism behind one of these cases will be named. A specific biological process, confirmed in peer-reviewed literature, that rewrote the ceiling of what vertebrate tissue can physically endure. The document exists. The researcher is named. The number is precise. The obvious explanation fits most of the story, most of it. When an animal survives extreme cold, extreme starvation, extreme oxygen deprivation, the first answer science reaches for is torpor, slowed metabolism. The body turns down its own thermostat. Heart rate drops, breathing shallows, everything continues at a whisper. A hibernating bear breathes once per minute. Its heart beats 8 times per minute. Slow but present, running. That model works for bears, for ground squirrels, for most of what we call hibernation. But Kenneth Story at Carleton University started measuring wood frogs in 1982, and what he recorded did not fit the model, not slowed, absent. No heartbeat, no respiration, no measurable neural activity. The instruments registered the same values they register when pointed at a stone. John Costanzo's team at Miami University, Ohio, pushed the measurement further. In a 2013 study published in the Journal of Experimental Biology, they confirmed frogs surviving at -16 degrees Celsius for multiple weeks. The cardiac arrest wasn't a brief interruption, it lasted for the entire frozen period. This is not torpor. Torpor is a dial turned low. This is the dial removed entirely. Then there is the Olm, Proteus anguinus, a blind cave salamander found in the underground rivers of the Balkans. A 2010 study published in Animal Biology documented individual specimens going without food for 10 years, not weeks, a decade. The researchers recorded metabolic rates so depressed they approached the threshold of detection. Jaime Bosch at the National Museum of Natural Sciences in Madrid studied the Olm's metabolic suppression and concluded the standard model of slowed but continuous function simply could not account for what these animals were doing. The framework breaks in 2 places simultaneously. 2 different animals, 2 different mechanisms, the same impossible result. Which forces a harder question. If the body isn't running slowly, what is actually keeping it intact? Here is the mechanism, and it is stranger than the survival itself. When a wood frog's skin makes first contact with ice, the liver responds within minutes. Not hours, minutes. It floods the bloodstream with glucose, a sugar concentration so extreme it would be toxic in any other context. Story and Story documented this precisely in their 2013 work. The glucose moves into cells, displacing water, occupying the space where ice crystals would otherwise form and rupture the cell wall from inside. The cells do not freeze. They are packed too tightly with sugar to freeze. The water between cells turns to ice. The frog's body becomes structurally a biological slush, and it survives. That is one animal. Now consider a completely different kingdom, a completely different mechanism, and the same brutal logic underneath both. The sea cucumber, Holothuria glaberrima, responds to stress by ejecting its own organs through its body wall, deliberately. Garcia Arraraz and Greenberg documented this in the Journal of Experimental Zoology in 1998. The digestive tract, the respiratory tree, the gonads, expelled entirely. The animal sits there, hollow, and then, within weeks, it rebuilds everything. A 2007 study in the Journal of Experimental Marine Biology and Ecology tracked the regrowth timeline. Measurable organ tissue at 2 weeks, functional restoration approaching 6. 2 animals. One dismantles its chemistry, one dismantles its anatomy. Both survive by becoming temporarily less than themselves. But here is where the wood frog story breaks open into something unresolved. The glucose flood is triggered by the first ice crystal touching the skin. That signal travels from skin to liver and back faster than the nervous system conducts impulses at freezing temperatures. The nervous system, at that temperature, is too slow. So what carries the signal? That question does not have a clean answer. The researchers who mapped the glucose response are the same ones asking it. Here is what the 2014 cryobiology paper actually said. Not what the frog does when it freezes, what the ice does. The research team at Carleton University found that the nucleation signal, the trigger for the entire survival cascade, does not travel through nerves. It travels as the ice itself, a mechanical wave propagating inward from the skin's surface. The body does not detect freezing and then respond. The freezing is the message. That distinction took years to establish. Once it was established, it broke something fundamental about how survival biology was understood. When ice contacts the frog's outer skin, a biochemical cascade fires. Glucose floods outward from the liver, rushing toward every cell in the body. That flood moves faster than the ice front. The cells are saturated with cryoprotectant before the crystals arrive, not because the nervous system sent an alarm, but because the physics of nucleation itself is faster than nerve conduction at near zero temperatures. The ice is not the threat the body is managing. The ice is the clock the body is running against, and also the starter pistol. But the Carleton team tested what happens when you remove the ice contact entirely. Same temperature, same frog, no nucleating surface. The cascade does not fire. The animal dies. Not from the cold, from the absence of ice. The very thing that kills every other vertebrate on Earth is the only thing that activates the mechanism keeping this one alive. Without the ice to trigger it, the survival chemistry never starts. The frog needs to freeze in order to survive freezing. Every other vertebrate tested at equivalent temperatures, no ice contact, same outcome, non-viable. The frog's solution is not to resist the ice but to require it. Which raises a harder question. If the frog can survive up to 65% of its body water converted to ice, what exactly is left that is still alive? Here is what was promised at the start. A vertebrate, common across North America, filmed, named, alive right now in forests from Alaska to Georgia. Up to 65% of its total body water converts to ice. Its heart stops. Breathing stops. Blood stops moving entirely. By every clinical definition applied to any other vertebrate on Earth, it is dead, and then spring arrives. The wood frog Rana sylvatica does not slow down when it freezes. It does not enter some suspended twilight. It crosses every threshold medicine uses to pronounce death. No pulse, no respiration, no measurable neural activity. It holds there for up to 8 months. In 2013, a study published in the Journal of Experimental Biology confirmed the mechanism fully. Up to 65% of total body water converted to ice. That number had been approached before, but confirmed at that threshold. In a vertebrate, it rewrote the assumption. The assumption was simple. Ice inside vertebrate cells destroys them. Crystals rupture membranes, cells die, the animal dies. Every clinical model said so. Every organ preservation protocol was built on that ceiling. The frog's liver ignores the ceiling. Within minutes of skin contact with ice, the liver floods the bloodstream with glucose. Not slowly, not gradually, a surge. That glucose enters the cells and displaces water before ice can form inside them. Ice forms between the cells, not through them. The distinction is everything. Outside the cell wall, ice is survivable. Inside, it is not. The frog found the only door through a wall biology said didn't have one. John Costanzo's team at Miami University documented frogs surviving temperatures reaching -16 degrees Celsius across multiple weeks. Cardiac arrest the entire duration. 11 minutes after thaw, a heartbeat returned. Not hours, 11 minutes. Cryomedicine researchers took note. A passage in a subsequent preservation journal named Rana Sylvatica directly as the model for endogenous glucose cryoprotection research. The frog that dies in a frozen Ohio forest every November is now informing how human organs might survive transplant waiting times measured in days rather than hours. The ceiling on vertebrate survival is not where biology said it was. One common frog, one confirmed mechanism, one published revision. The question of what a living thing can endure had to be asked again from the beginning. This record is filed. The archive remains open.